Where does energy come from?
It comes from glycolysis.
What is glycolysis?
Glycolysis is the breakdown of glucose to extract energy for cellular metabolism.
The first step of glycolysis is conversion of D-glucose to glucose 6-phosphate, which catalyzed by hexokinase.
The second reaction of glycolysis is the rearrangement of glucose 6-phosphate into fructose 6-phosphate by glucose phosphate isomerase.
Phosphofructokinase with magnesium as cofactor changes fructose 6-phosphate into fructose 1,6-bisphosphate.
Adylase splits fructose 1,6-bisphosphate into two sugar that are isomer to each other, which are dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. Triose phosphate isomerase rapidly interconvert the molecule DHAP and GAP. Glyceraldehyde phosphate is removed or used in next step of glycolysis.
Glyceraldehyde 3-phosphate dehydrogenates and adds an inorganic phosphate to glyceraldehyde 3-phosphate, producing 1,3-bisphosphoglycerate.
Phosphoglycerate kinase transfers a phosphate group from 1,3-bisphosphoglycerate to ADP to form ATP and 3-phosphoglycerate. The enzyme phosphoglyceromutase relocates the P from 3-phosphoglycerate from the third carbon to the second carbon to form 2-phosphog. The enzyme enolase removes a molecule of water from 2-phosphoglycerate to form phosphenolpyruvic acid PEP. The enzyme pyruvate kinase a P from PEP to ADP to form pyruvic acid and ATP.
Kreb cycles, also known as citric acid cycle, it is a series of chemical reactions used by all aerobic organisms to release stored energy through the oxidation of acetyl coenzyme A derived from carbohydrates, fats, and proteins. Used to generate energy through oxidation of acetyl coenzyme A. Synthesis of NADH and production of amino acids. The first reaction is the formation of citrate. The first reaction of the cycle is the condensation of acetyl coenzyme A with oxaloacetate to form citrate, catalyzed by citrate synthase. The second reaction is the formation of isocitrate. The citrate is rearranged to form an isomeric form, isocitrate, by an enzyme acontinase. In this reaction, a water molecule is removed. Reaction 3 is oxidation of isocitrate to alpha-ketoglutarate. In this step, isocitrate dehydrogenase catalyzes oxidative decarboxylation of isocitrate to form alpha-ketoglutarate. In the reaction, generation of NADH from NAD is seen. Reaction 4 is the oxidation of alpha-ketoglutarate to succinyl's coenzyme A. Alpha-ketoglutarate is oxidized. During this oxidation, carbon dioxide is removed, and during the oxidation, NAD+ is reduced to NADH and H+. The enzyme that catalyzes this reaction is alpha-ketoglutarate dehydrogenase. Reaction 5 is the conversion of succinyl coenzyme A to succinate. Coenzyme A is removed from succinyl coenzyme A to produce succinate. The energy released is used to make guanosine triphosphate GTP from guanosine diphosphate GTP and pi by substrate level phosphorylation. The enzyme is succinyl coenzyme A synthase. The GTP can then be used to make ATP. Reaction 6 is the oxidation of succinate to fumarate. Succinate is oxidized to fumarate. During this oxidation, FAD is reduced to FADH2. The enzyme is succinate dehydrogenase that removes two hydrogens from succinate. Reaction 7 is the hydration of fumarate to malate. The reversible hydration of fumarate to malate is catalyzed by fumarate of fumarate hydratase. Fumarate continues the rearrangement by adding hydrogen and oxygen back to the substrate. Reaction 8 is the oxidation of malate to oxaloacetate. Malate is oxidized to produce oxaloacetate, which is the starting compound of the citric acid cycle by malate dehydrogenase. During this oxidation, NAD+ is reduced to NADH and H+.
The mitochondria has an outer mitochondrial membrane, an inner mitochondrial membrane, and the space between these two membranes is called the intermembrane space. And inside we have the matrix of the mitochondria. Now, if we look closer at the section of the membrane structure of the mitochondria, we can see the structure of the electron transport chain. So here we have the outer mitochondrial membrane, the intermembrane space, the inner mitochondrial membrane, and the matrix. The inner mitochondrial membranes contains 4 sets of enzyme complexes which are named from 1 to 4 in a Roman numerals. The matrix of the mitochondria contains numerous other cycles like Kreb cycles and beta oxidation. It is mentioned in the previous video that these cycles produce reducing equivalents called NADH and FADH2. These reducing equivalents contain electrons which are donated to this set of enzyme complexes. The electron then transferred from the first to the fourth enzyme complex, and in this movement, energy is generated. This energy is utilized in the pumping of hydrogen ions into the intermembrane space from the matrix of the mitochondria. This continuous pumping of hydrogen ions into the intermembrane space causes the generation of a higher concentration of hydrogen ions in the intermembrane space as compared to the matrix of the mitochondria. This generates a positive charge in the intermembrane space and negative charge in the matrix of the mitochondria. This is called the electrochemical gradient. The hydrogen ions cannot cross against this electrochemical gradient because the intermembrane space is non-permeable to ions. So here we require a special transporter, the enzyme ATP synthase. The ATP synthase transports hydrogen ions into the matrix of the mitochondria, and it uses the energy generated from the flow of hydrogen ions to phosphorylate adenosine diphosphate into adenosine triphosphate. And this is the basis of the generation of energy in the form of ATP, which is a currency of energy in our body.